Okay, so welcome to this next video in which we're discussing the adaptive immune response and immunosuppressants. Okay, so we're in the process of discussing the adaptive immune response. We've discussed that we're going to divide the adaptive immune response into three separate reactions. One that is to humoral or extracellular pathogens, and then two which are to intracellular pathogens. One that is directed at cytoplasmic intracellular pathogens, and one that's directed at these intracellular pathogens that are within phagosomes. Okay, so we're going to start with the humoral or extracellular pathogens. Okay, so before we start, we need to discuss something which is quite important, which is the concept of an antigen. Okay, so an antigen is not a difficult concept. It is just any protein which is capable of having an antibody made against it. Okay, now we're going to divide antigens into two major sorts. Okay, so antigens which are on the surface of the pathogen. Okay, so these are prime targets for antibodies to be made against them. Okay, so here is our pathogen again, in pink here, and basically it will be covered in a whole bunch of proteins which are very, very different from any protein that would ever be put on a healthy, normal human cell. And this is very important, that antigens are also proteins that human cells would never ever make. So the target for the adaptive immune response, or at least the humoral adaptive immune response, is going to be these antigens that cells would never ever make. So we are going to try and kill these pathogenic cells, and we need to recognize these pathogenic cells. And the way we're going to do it is by looking for proteins on the surface of these pathogenic cells which would never ever be on a normal human cell. Okay, so these are the proteins that we are going to produce antibodies against. So one of the types of antigen are these antigens that are on the surface of cells. Another type of antigen that we might want to make an antibody against is a secreted antigen. So if the microbe is actually producing some protein that no human cell would ever produce, and potentially this is a toxin, this is something that's going to go and attack normal human cells. Okay, this would be a very good target for antibodies, because if an antibody binds to this, it will neutralize it basically, and it will stop the toxin from then being able to harm other human cells. So basically, the humoral immune response is going to be all about these antigens, these proteins that are either on the surface of the cell or that are being secreted from the cell. And if they're being secreted from the cell, then potentially they're a toxin, and therefore we want to attack that toxin to stop it from working. Or if it's on the surface of the cell, we can use that for a recognition purpose, basically. So there are these two major types of antigens that we're going to launch a humoral immune response against: surface antigens and toxins. And the point of that response is slightly different. The point for the surface antigens is so that we can recognize and attack the microbe. The point for the secreted toxins is to neutralize them. Okay, so that's the concept of an antigen, and we've got these two types of antigen now. So let's begin then the humoral immune response. So it begins with something known as a professional antigen presenting cell. Okay, so we're going to start with a professional antigen presenting cell. Now, what these cells are going to do is they're going to phagocytose the extracellular pathogen into a phagosome, and they're going to break that pathogen down along with all of its proteins, and they're then going to process these antigens that are on the surface of the pathogen, or potentially the antigens that are being secreted from the pathogen. So let me explain. Professional antigen presenting cells. Okay, and these sort of cells are often abbreviated to APCs for short. Okay, So these cells may well phagocytose this pathogen, or they may phagocytose the secreted toxin potentially. So what will happen is the cell will phagocytose the pathogen, so it will end up with the pathogen inside of it in a phagosome. So this is the nucleus of the professional antigen presenting cell, and then let's let this be the phagosome. And we'll imagine that we're going to launch an immune response against the antigen that's on the surface, but it's a very similar process for the secreted one as well. Okay, so here is the pathogen here, and we'll colour it in pink. And it's got this nice antigen, and we're going to mount an immune response against this specific antigen that I've drawn. Now, of course, a pathogen is not necessarily just going to have one antigen that you're capable of launching an immune response against. So there may be hundreds of antigens that you're going to actually launch humoral immune responses against. So this is simplified. We're just going to look at one. Okay, so it's going to phagocytose the pathogen. It's then going to break it down by fusing the lysosomes with the phagosome and releasing these lysozymes into the phagosome, and that will break down the microbe. In addition, it will break down this surface antigen here, and you'll get loads of fragments from this surface antigen. Okay, so remember, the surface antigen is a protein. Antigens are always proteins. So we'll break the protein down, not quite into amino acids, but we'll break it down into little fragments. And we're now going to present these fragments on a protein complex that is on the surface of these cells. Okay, so this protein complex is what's known as Major Histocompatibility Complex Class 2. So this is the Major Histocompatibility Complex Class 2. So this is, because Major Histocompatibility Complex Class 2 is a bit of a mouthful, this is often just abbreviated to its initials, which are MHC, and then you put Roman numerals 2. So M for major, H for histocompatibility, C for complex, and then just a 2 like that with a dash in between the two. Okay, so MHC Class 2. So whenever, and this is really, really important, this is going to be really important later, whenever you have an antigen that you got from a phagosome, basically, okay, or whenever you have an antigen fragment that you got from a phagosome, you present that antigen fragment on Major Histocompatibility Class 2. So it's broken down, this antigen that it has got within its phagosome, it's broken it down into fragments, and it will put these fragments on Major Histocompatibility Complex Class 2. So the whole antigen doesn't get put here, just a little fragment of it, okay? Now, I'm going to have to continue drawing this as a little blue box, but don't think that that represents the entire antigen, even though the entire antigen was represented as a little blue box. This now is just an antigen fragment, and by the way, the abbreviation for antigen is to write AG like so. So this means antigen and then fragment. Okay. So, whenever you have an antigen which is within a phagosome and you're breaking it down into fragments, those fragments will be put on your surface on MHC Class 2. That is important. Whenever it comes from a phagosome, it goes on to MHC Class 2, not MHC Class 1. If it comes from the cytoplasm, it goes on to MHC Class 1, and that will be important when we look at the cytoplasmic pathogens later. Okay, so let's now talk about what sort of cells are professional antigen presenting cells. Okay, so examples of professional antigen presenting cells, the two key examples that are going to kickstart the entire adaptive immune response are dendritic cells. Okay, these are kind of the archetypal one. These are the ones which really do what we're about to describe, okay? And they sort of look a little bit like the cell bodies of neurons. So they have processes
coming off their central cell body here. So here are the processes coming off the central cell body. Okay, so they have dendrites basically, and here is the nucleus. Now they have absolutely nothing to do with neurons, even though they look slightly like a neuron. They are not capable of conducting action potential, so they're non-excitable. They simply don't have the voltage-gated channels that you would need in order to conduct an action potential. Okay, in addition, another professional antigen presenting cell is also a macrophage. Okay, however, macrophages aren't really that keen on doing it. They can do it, but they don't do it nearly as much as dendritic cells. Dendritic cells are fantastic at doing it. Now, B cells are also counted as a professional antigen presenting cell, but generally they don't kickstart the humoral immune response. Generally it's either a dendritic cell or a macrophage which is going to kickstart the adaptive humoral immune response. Okay, so let's just quickly talk about what the difference between a normal antigen presenting cell and a professional antigen presenting cell is. So basically all cells in your body which are nucleated can present antigens. So all cells in the body have proteins in their cytoplasm, and they continually chop up proteins that are in the cytoplasm, degrade them down, and then stick fragments of them on their surface bound to major histocompatibility complex class 1 molecules, because if it comes from the cytoplasm it gets put on MHC class 1. Okay, so all cells can antigen present. So you might think, oh, so is the difference between professional antigen presenting cells and non-professional antigen presenting cells, or just normal cells, because all cells are capable of presenting antigens except non-nucleated ones such as erythrocytes. Okay, is the difference that these ones have MHC class 2 and it's not? The difference between a professional antigen presenting cell and just a normal cell, and I would just like to stress that when people say antigen presenting cell, they mean a professional antigen presenting cell. So you wouldn't describe a normal cell as an antigen presenting cell, okay? You would just say that it does antigen presentation, but it's not, its main role is not antigen presentation. Okay, its main role is maybe, you know, it could be a liver cell, so its role is in metabolism. Okay, the main difference between these professional antigen presenting cells and a normal cell is that they have the ability to stimulate T cells and normal cells do not. And the reason they have the ability to stimulate T cells is that they're not just going to put antigen fragments on their surface with MHC class 2. They're also going to put a whole bunch of other molecules that are needed to activate T cells, and we'll talk about these later, and I'll come back to this point when we do discuss those other molecules. Okay, so what's happened so far? The pathogen invaded our tissue. Okay, then dendritic cells and macrophages, and dendritic cells were already present they're all over your body, so all over your tissues you'll have dendritic cells interdispersed around. Macrophages will have flooded into the affected tissue as part of the acute inflammatory response, and you also do have a few resident macrophages which are dotted around all tissues just like dendritic cells. So when the acute inflammatory response happens, you've got loads of these macrophages and some dendritic cells already there, and these will be phagocytosing pathogens, basically, that are in this infected tissue. Okay, and when they phagocytose them, they'll then break them down and put antigen fragments on their surface. However, they now need to get to T cells because they're going to use this to activate the adaptive immune response. Specifically, we're talking about the humoral adaptive immune response. So now what's going to happen is this professional antigen presenting cell is going to go to where T cells are present. So where do T cells hang out? Well, they hang out in lymph nodes. So this professional antigen presenting cell is going to go to the lymph nodes. So all tissues of your body have a direct communication line to lymph nodes. They have lymph vessels which drain them. Okay, so here is a lymph vessel. Okay, so this is a lymph vessel, and it's going to drain our infected tissue, and it's going to drain interstitial fluid from the affected tissue to regional lymph nodes. Okay, so here we have our lymph nodes, and let's just go over the structure of a lymph node because it's quite important for understanding. Okay, so lymph nodes have a huge number of lymph vessels coming into them. So they have a large number of afferent lymph vessels. That's a good word. Let me write that down. Afferent refers to lymph vessels which are coming into the lymph node. So this is an afferent lymph vessel. All of these vessels that I've drawn so far coming in are afferent lymph vessels. Okay, then the lymph node will have one bigger lymph vessel coming out. So it only has a single efferent lymph vessel. So efferent means coming out. Okay, so this is an efferent lymph vessel. Okay, so let me make this crystal clear by putting on some arrows. So this one will be draining lymph in this direction out of the lymph node, and these will be draining lymph in this direction into the lymph node. Okay, now when lymph arrives in the lymph node, firstly it goes into a space known as the subcapsular sinus. So all around the periphery of the lymph node there is a portion known as the subcapsular sinus. Okay, so this is basically just continuous with the afferent lymph nodes, afferent lymph vessels coming in. So this is the subcapsular sinus. And it's called that because it's just underneath the capsule of the lymph node, which is this outer boundary of the lymph node. And also it's a space that's full of fluid, and spaces which are full of fluid are called sinuses. Okay, then underneath the subcapsular sinus, what you have is the start of the solid matter of the lymph node, basically, because this obviously is all the lymph fluid in the subcapsular sinus. Underneath what you have is the start of the solid material. Okay, now what you have firstly is a region known as the cortex of the lymph node. So this, which I've outlined now, is the cortex of the lymph node. And this basically is absolutely full of naive T lymphocytes. Okay, so packed into this entire space, you have just got a huge number of naive T lymphocytes. And I should just explain something about naive T lymphocytes. So, naive means that they have never been activated before, okay? So they are sitting and waiting. Now there are two main divisions of naive T lymphocytes. There are two main populations. There are the CD4 positive naive T lymphocytes, and there are also the CD8 positive naive T lymphocytes. Okay, so let me divide these two up. So CD4 positive naive T lymphocytes look like this. So all lymphocytes have a massive great nucleus. Okay, well actually it's not that the nucleus is massive, it's just that the cytoplasm is tiny. Okay, so because they're very inactive cells, they don't need much cytoplasm because that's where all the chemical reactions happen. If they're doing nothing because they're just sitting there for all eternity waiting to be activated, then they don't need a big cytoplasm. Okay, now CD4 positive T lymphocytes have on their surface a molecule known as CD4, which stands for cluster of differentiation or cluster of designation 4. So let me colour this in one colour. So we'll have this in pink here. Okay, whereas CD8 positive naive T lymphocytes have a different molecule on their surface. Instead, they have this molecule CD8. So here is the nucleus again, taking up most of the cell. Okay, and now on the surface of these CD8 positive naive T lymphocytes, you instead have CD8. Now, T lymphocytes, mature T lymphocytes, do not have both CD4 or and CD8. They have one or the other. They either have CD4 or they have CD8. There's no breaking of that rule. You have one or the other. You do not have both. Okay, so in the cortex you have a huge number of T lymphocytes. Loads of them will be CD4 and loads will be CD8. Okay, so the ones that are going to be important for the humoral immune response is these CD4 positive naive T lymphocytes. Okay, now if we ignore the half of the T lymphocytes which are CD8 positive T lymphocytes and then just look at our population of CD4 positive T lymphocytes, you will have a plethora of different T lymphocytes. No two T lymphocytes will be the same. Now, how do they vary? Well, it's not in how they look underneath the microscope. They all look pretty much identical under the microscope. But all of the T lymphocytes have something known as a T cell receptor, which I'm drawing now here. Okay, so this is going to represent our T cell receptor. Okay, and T cell receptor is often abbreviated to TCR for short. Okay, so all T lymphocytes, all naive CD4 positive T lymphocytes have a T cell receptor. Okay, and this T cell receptor will be different in the case of each naive CD4 positive T lymphocytes. So if you go along looking at different naive CD4 positive T lymphocytes, you'll find that they all have slightly different T cell receptors. So this is how they're all different. And all of these T cell receptors recognize a certain antigen fragment. Okay, so they'll bind to certain antigen fragments and not to others. So all of them are primed against, they will fight against a certain antigen fragment. Okay, so it is these T lymphocytes that we are going to present our antigen fragment to when it's on MHC Class 2. Okay, so let's just finish the structure of the lymph node now. Also in the cortex of the lymph node you do have these spheres or follicles which are not T lymphocytes. Okay, so you have follicles within the cortex which are not made up of T lymphocytes and are instead made up of B lymphocytes. So these are what are known as the B cell follicles. Okay, so they're not filled with T lymphocytes, but instead are filled with B lymphocytes in there, and we'll come back to B lymphocytes later. Basically, B lymphocytes, again, there isn't the two distinct populations as there were with naive T lymphocytes. Oh, and I should have added that. These are full of naive B lymphocytes, so B lymphocytes that have never ever been activated. Okay, now B lymphocytes, again, they all vary basically, they're all different. They all have a slightly different B cell receptor this time, which recognizes a slightly different antigen, but we'll talk about that later. Okay, then there is another layer of the lymph node. Underneath the cortex there is another layer which has these sort of invagination, or evaginations almost, projections into the center here. And this is what's known as the paracortex of the lymph node. So this other lower down there here. This is the paracortex. Okay, so let me colour in a few things here to make it simpler. So, in red here, this is the cortex where the T lymphocytes are. Okay. Then we have these B cell follicles, which I'll colour in green. Okay, so I'll give them a little hint of green there. Okay, then we have the paracortex, which again I think I'll colour it in a different colour, but I'll colour it in pink here. Okay, so this is the paracortex. Okay, and this again consists of T lymphocytes basically, so again it's absolutely full of naive T lymphocytes of both the CD4 type and the CD8 type. Then right at the centre you have this medullary sinus, as it's called, and this is full again with lymph fluid. Okay, so this is the medullary sinus. So basically it's a very clever structure because the lymph comes in and goes into this subcapsular sinus, then it has to somehow get to the medullary sinus, and the only way it can get from the subcapsular sinus to the medullary sinus is to trickle through the cortex and the paracortex, basically. So all of the lymph has to pass through this mass of T lymphocytes and B lymphocytes, and this is how lymph nodes function. They are watching, they are checking all of the lymph fluid that is coming to them, and they are checking it for signs of infection, basically. So they are watching, they are checking that the tissue is not infected, and that's how these lymph nodes are going to function. Now, what's going to happen is that our professional antigen-presenting cell, whether it is a dendritic cell or a macrophage, is going to come into the lymph node through one of these afferent lymph vessels, and then it's going to have to percolate through the layers of lymphocytes, and it's going in that process to meet a huge number of T lymphocytes. Now, it needs to find a T lymphocyte which has a perfect T cell receptor for the antigen fragment that it has posed in MHC Class 2 on its surface. Okay, and we'll continue this discussion in the next video.